we have started with our opening remarks, and so hopefully this has helped to set the stage for today's conversation. We're going to have two main sessions within the symposium for today. And as you just heard, we do have 4 different scales at which we are considering research needs related to pandemic prediction. And so our first session is going to focus on the big picture, the end-to-end scale. And this session is going to be moderated by Dr. Prakash and Dr. Wigington. During the session, we'll have a number of keynote and supplementary presentations, as well as a panel Q&A discussion. Later, there will be a breakout session on this particular topic. Now, many of you have received individual links via email to be able to participate in those breakout sessions. After the breakout, there'll be some group discussion, as these breakout sessions will be happening in smaller parallel groups. Having smaller conversations will then come together in the report outs to get a better feel for the conversations that took place. After this, there will be a break for lunch. If you are only participating in the webinar and not in the breakout sessions, then after our panel Q&A, you will be able to rejoin us again here in the webinar after the lunch break. Following session 1, we will have a discussion on our second theme, looking at research needs at the molecular level. And we'll have a very similar flow: our keynotes, our follow-up presentations, the panel Q&A, and then the breakout discussions, which will again be happening using a different platform. Those of you who are participating will have already received those links via email. So after the report outs, we will come back together again here in the Zoom webinar for some closing discussion. All right, so we look forward to all of your participation today during these 2 different discussions. And again, if you have questions at any point, please provide those questions in the chat box because they will inform the discussion that we have during the Q&A. So we are going to begin now with our plenary talks. And so again, our first session is at the end-to-end level. And so at this point, what I'd like to do is turn the agenda over to our session chairs, Dr. Prakash and Dr. Wigington, who will introduce our plenary presenters and lead us through our panel discussions.
Thanks a lot, Emmanuel. So for those who are joining us just now, so we'll be starting with the end-to-end theme of this workshop. So we have a set of really great 3 speakers who will be giving talks, and then there will be a short panel at Q&A where we welcome the audience to enter their questions in the chat. So as our plenary speaker, I'm really delighted to introduce Professor Madhav Marathe from the University of Virginia. So Madhav is an endowed distinguished professor in biocomplexity, director of the Network System Science and Advanced Computing Division, the Biocomplexity Institute and Initiative, and a tenured professor of computer science at the University of Virginia. Dr. Marathe is a passionate advocate and practitioner of transdisciplinary team science. During his 25-year professional career, he has established and led a number of large transdisciplinary projects and groups. His areas of expertise are network science, AI, HPC, computational epidemiology, biological and socially coupled systems, and data analytics. So I won't go into details of all the awards Madhav has won throughout his long career, but I would just like to highlight that you can see that he's the fellow of almost all societies you can think about. So we are really fortunate to have him to help us get off to a strong start. With that, over to you, Madhav. Thank you.
Thanks so much. Can I share my screen to do that? Aditya?
Sure, Madhav. Yep.
Okay.
But we can't see it yet. Yeah.
Yeah. Okay.
Yep.
You can see it now, right?
Yes. Thank you.
Okay. Thank you very much, Aditya. It's a pleasure and honor to speak at this meeting. It's second of the 4 meetings that Mitra described, and I'm really grateful for being given the opportunity to talk here. Just before I start, I want to point out that there is a virtual organization that NSF just funded called Prepare. I have put in the link here, and the goal is to really get folks interested in this general topic to come together and find common collaborative points of interest so that we can advance this topical area. Um, Let me thank our team members at the Biocomplexity Institute, our collaborators at various universities, um, for working with us very closely. This work is certainly not my work alone, but in fact, my portion is relatively small at this point of time. Others have done some amazing work in pulling this program together. I also want to thank various funding agencies, including DoD, NIH, CDC, but importantly, NSF, who has been very kind in terms of providing funds to us in carrying out the work we've done. So I'm going to break down my talk into 3 parts. The first part will focus on the roles of models and decision making. And I'll conclude that part by providing some examples of successful application of models during the COVID-19 outbreak. In the second part of my talk, I will outline some of the challenges that we have all faced while we support epidemiologists in the field. And many of you have already talked about these aspects, so I might be brief in this particular part of my talk. And finally, I'll describe the work that our group has done along with our collaborators over the last year in terms of planning and responding to the COVID-19 outbreak. And then I'll conclude with final thoughts and takeaway messages then. So let me start with why we would like to model in the first place. And the view that I have is that models have to go beyond just being useful for predictions. In real time, models that support computational epidemiology should be able to synthesize available data. They should be able to provide a range of interpretations to incoming measurements, evaluate range of options and outcomes from various response strategies, monitor the effect of these response strategies, coordinate among various stakeholders, and finally, they should be made useful not just by computing experts, but by analysts. And this aspect of modeling really goes beyond prediction, and hopefully this will become clear throughout my talk as I talk about end-to-end modeling and prediction here. So from a decision-making standpoint, we view individuals as agents, of course, and they follow this sense, interpret, deliberate, act loop that I've shown here in a simple picture. But as you start using and thinking about models in the real world, such as right now, in the case of COVID-19, you realize that you have multiple stakeholders, multiple types of decision makers. You have citizens, daily citizens doing their day-to-day job, authorities, local, state, and federal authorities, and then on-the-ground responders, just to name a few of these classes. And each of them tries and makes decisions by either building a mental model or using a computational or mathematical model in a form that is such that models might not be compatible in a typical form that we are so used to. But hopefully they provide context-specific information and decision-making capability to each of these decision makers. And this view is really important as we study and build models for decision-making to support epidemic science. And in fact, I would argue for other complex systems as well. So let me give you a few examples of successful modeling efforts by other folks in the community in the last 1 year or so. This is a very simple cartoon of how the COVID-19 outbreak evolved in time. And I've broken down time just for the sake of simplification into 4 phases. The first phase that starts end of February or so goes on to June. Second phase goes from June to September. The third phase goes from September to January, and then we are right now in the 4th phase. And the thought is that each of the phases actually allowed us to study different questions. Different questions were posed by policymakers, individual citizens, first responders, and the models were used as a result in different forms. So in phase one, for instance, and this is an incomplete list without a doubt, right? But hopefully you'll get a sense for how models get used and what work others did over this period of time. So in the first phase, there was clearly a question that was pertaining to risk of importation of disease from China into other parts of the world. There was questions related to inferring disease parameters, and we'll hear a little bit about it today as well. And we wanted to understand the impacts of social distancing. And models from Imperial College, IHME, Northeastern, and Columbia did an amazing job, in my opinion, on the whole, to try and answer these questions. In the second phase, we sort of pivoted into new set of questions along with the old questions, of course. Questions relate to risk of resurgence. We had seen the pandemic really come down in the US and many parts of the world, Europe included. So we wanted to understand, as we open up the society, what sort of resurgence possibilities would we have? What is the possibility of using contact tracing apps? And again, here, work by UMass and others as a part of the ensemble, the use of surveys that were set up by CMU and Facebook, resource allocation work that Yale and UT Austin did, and testing work that the African Institute of Mathematics did, played a very important role. In the third phase, we started to see the epidemic having a resurgence. We started to understand the effect of closing schools, colleges, or opening them, and how we would go ahead and open them, understand and predict the use of medical resources, as well as understand the effect of seasonality. The work done by Oxford Group, the work done by Princeton Group, Brian Grenfell, who is going to give a talk tomorrow, I believe, and the work done by my colleague Yuri Leskovic at Stanford, all did some amazing work again in this area, supporting policymakers as they went along. And finally, we are in the fourth phase, which is when we are trying to understand variations in the virus itself, and trying to understand the effect of vaccine rollouts. And work by Scripps and Colorado and Harvard stands out in this context. So let me move on to the challenges in the second part of my talk. But before I do that, let me point out that while we had a lot of success, we also fell probably short in some ways. We were not able to forecast the disease dynamics that accurately in many cases, although we did quite well. We were somewhat surprised to the low usage of digital contact tracing apps. We learned that interventions often do not work, even though they are meant with good intentions, because of lack of compliance and potential misinformation. So these challenges continue to, of course, be in front of us, and hopefully in the next round, we'll be able to say a little bit more about it. So let me move on to the set of challenges that we face from an end-to-end perspective. And I've broken it down into about 4 different class of challenges. The first one, which was covered quite well in the first workshop that was carried out last week, it talked about zoonosis, talked about viral evolution, talked about immunological response. And the main theme that I want to present as a part of this slide as a result is that a single model is not going to help us solve this end-to-end problem. We need to find ways to couple models of virus evolution, the human immune system, to epidemic spread and socioeconomic systems. There's another aspect of end-to-end that is worth noting, which is scales. There's spatial scale that goes from a household all the way to a country and to the globe. There's a temporal scale that goes from minutes and even smaller time scales to years, in which the virus and the society evolves. There is a social scale in terms of interactions between members of the household, to the community, to the county, to the state, to the country, and to the globe. And finally, there is issues that have to relate process of within-host disease transmission, individual behavior change, community response, global response coordination, and so on and so forth. Here, as a result, I would argue that multi-scale, multi-theory, and multi-level network representations and models are absolutely the key. The third part of end-to-end modeling and informatics has to do with trying to take data and converting them into decisions and communicating these ideas effectively to policymakers. This figure is maybe too crowded, but the basic idea that I would like to convey is that one gets a large variety of data from different data sources. Issues about IP, issues about privacy, issues about commercial use, issues about confidentiality, all of them play a big role in access to these data sets. The models need to work closely with the data sets that are available, produce meaningful insights, and then provide them to the decision makers as they make decisions using these data sets. And the last topic I would like to cover, which always gets asked to a modeler, is one of validation and uncertainty quantification. In the end, all of these models, as we all know from the famous quotes, are wrong, but I think they are useful in many cases. And I think if you want to make the models for epidemic science to be useful, I would argue that notion of validity has to be extended away from the standard notion of retrospective and predictive validity, and to talk about internal structural validity, as well as understanding the role of models to see when certain assumptions are failing to capture real world dynamics and then adapting them. There are other challenging questions related to governance, ethics, logistics, diplomacy, and One Health that all are important, but I won't cover it in today's talk. So with that, the time I have, let me try and quickly outline the program that we have built here and that we used as a vehicle to support various authorities during the last 1 year as we worked closely with them as they responded to the COVID-19 outbreak. So the way we have approached this problem is this concept of networked epidemiology. We did this work early on back in 2005 in our Nature paper, where we showed how networks can meaningfully represent in silico social contacts and how those networks can then be used to derive important policy insights. We have taken that program quite a bit ahead, where now we are able to synthesize these networks using very detailed data from digital traces, from surveys and such, and build very realistic social contact networks. We have just completed the first version of this social contact network for the entire US, and we are working towards improving it as we go along. In the second step, we use a social contact network. In fact, it's a multi-theory, multi-scale network. It has got aspects of disease transmission. It has got representation of individual and collective behaviors. We use models to try and simulate various contagion processes on top of these networks to study how diseases might propagate, how information might propagate, what sort of actions might be taken, and then use all these information sets and the results that the simulations produce to carry out counterfactual analysis, to study strategies from medical costs to predictions, and to do analysis of what might happen under certain scenarios. So,
Dr. Marathe, you have about 5 minutes left.
Sure. And I think there should be enough time. Thank you very much. So let me give you a practical perspective of the work we have done quickly, because I think this is a very, very satisfying aspect of the work we have done. For the last 1 year, we have been supporting multiple agencies and groups very actively as they respond to pandemics. I want to really acknowledge the work that Brian Lewis and Srini have done in trying to coordinate this on behalf of our group. The 80 of us have come together to try and do this week after week, and we are probably in the 52nd week right now of presentations to various agencies, from DoD to various parts of the Commonwealth of Virginia to our local agencies. This has been a very satisfying effort. We have learned how to try and do real-time epidemic science as we support important policy questions. What sort of updates and briefings do we give? We said we give about 6 to 7 briefings a week. And if you go to the state's Virginia Department of Health's webpage, you will see various reports that they put out based on the models and the data that we produce. Things which give here things like situation assessment, literature survey, model projections, narrative summaries, and such. Similar work is carried out for other agencies, but I'm going to focus on the work we have done for our state. It's easy to narrate one particular piece. For instance, every week we try and do projections. We try and study where the pandemic might go under different scenarios. And this notion of scenario analysis and projections has become more and more important rather than just doing predictions. We have built out various dashboards and tools. We built out the surveillance dashboards very early on. Johns Hopkins is a very nice dashboard as well, done around the same time. We built out other dashboard for national resource demand modeling for medical resources. With the group at Stanford, led by Michael Bernstein, we try and collect, using crowdsourced methods, various interventions and policy that were put in by counties in the US. And very recently, we worked with Yuri Leskovic's group to build a mobility impact dashboard based on a very important paper that he and his colleagues wrote in Nature. And these dashboards are all in use at various groups across the federal and the state government. We have also supported the CDC COVID-19 forecasting ensemble. We've been providing information to them every week for the last about 6 months or so. Aditya has been very active, has done very well in that program. But I want to again acknowledge Nick Reich's group and other forecasting methods, especially methods from Los Alamos, Nick's group, CMU, and others who have all participated in this to provide very meaningful forecasts to the various agencies that look at it. We have built a Bayesian ensemble ourselves. And what we have done is we have been able to build a multi-scale system where we can forecast the disease outcomes for the whole country, for the state of Virginia, and then every county in the country. So here, for example, I show the whole US outcome. We have shown outcome for Virginia, but we have the same basic map for every state in the country, and then every county in the country. So 3,500 counties are being studied in the models on a week-to-week basis. We've used these models to study a number of counterfactual analysis questions. One that I want to quickly outline is where we use the Pittsburgh Supercomputing Center, and thanks to NSF and Pittsburgh Supercomputing Center for providing this HPC access to us, we are able to compute and estimate the cost of COVID in terms of medical expenses. Another paper that talks about the bigger economic cost is also under review. And the interesting part here is that we had to really build HPC grid where computation was done on our local machines. Some aspects of computation was done nightly on the Pittsburgh Supercomputing Center. And then the data was brought back to our supercomputers here to glean insight from it. And one of the insights that we report in our Nature Scientific Reports paper is the medical costs, which we estimate to be $1 trillion, of course, in the worst case, but we show that interventions can actually lead to lower and lower medical costs. This is not surprising, of course, but the numbers turn out to be rather important and interesting. I'll conclude my talk in terms of results by this very recent exciting result that we have gotten. This paper is on medRxiv. You can take a look at it. What we did was we used the digital twin that we had built out to study vaccine prioritization strategies. And our results were quite exciting and interesting from our perspective. We showed that if you vaccinate individuals based on their contact time or degree in the social contact network, you can actually end up just in the state of Virginia and just in 2 months, 50,000 to 100,000 fewer infections, 700 to 1,000 fewer deaths, and 8 to 13% fewer hospitalizations. And if we extrapolate these results across US, we think this would have a significant impact. We think this is a really important strategy to consider, especially in resource-poor countries, where I do not think vaccines are going to be available anytime soon, or when they're available, they're going to be available in very small amounts. You know, the idea of using high-degree nodes to vaccinate is a folklore at some level. What is really new here is that nobody has ever studied this when the network is actually changing in time because of pharmaceutical interventions, because the vaccines being available not right at the start, but vaccines being available through a schedule, and all the behavioral adaptations individuals are doing as the epidemic progresses. The fact that this result holds even during this form makes the problem very interesting for us. So let me conclude with lessons learned from our work that we have done. This includes a couple of different lessons. One, we find that working closely with stakeholders and policymakers really helps us make models that are useful, explainable, transparent, and allows us to build better models. We found out that we need to be agile and flexible. I cannot overstate the importance of the transdisciplinary team science. This work could not have been done had we not assembled a team of this size and with so much expertise across the board. And finally, I think communicating scientific results in such situations needs to be very thoughtful and deliberate. So let me conclude my talk by saying that an effective strategy to reduce the global pandemic must detect the timing and location of occurrence, anticipate public reactions, and develop actionable interventions that enable targeted and effective response. I think this is not the first pandemic we are going to see in the next few years. I hope so. Hopefully not. We won't see many of them in our lifetimes for sure. But the looming challenges from climate change, antimicrobial resistance, synthetic pathogens, infodemics, urbanizations and global transactions, and expectations of timely information from everyone make addressing challenges that we might face when the next epidemic comes, which certainly will happen, to be something that we need to be prepared about. So with that, I will stop, and hopefully we can have a question answer session at the end of the 3 talks. Thank you.
Thanks a lot, Madhav, for that great talk. So I think I'll hand over the mic to my co-organizer, Krista, for the next speaker. Thanks.
All right, I'd like to introduce our next speaker, Dr. Debra Peters. Dr. Peters is an ecologist for the U.S. Department of Agriculture and the lead principal investigator for the Jornada Basin Long-Term Ecological Research Program in Las Cruces, New Mexico. Dr. Peters has greatly contributed to the broader research enterprise as senior advisor to the chief scientist at USDA and as a member of the National Ecological Observatory Network's board of directors. In addition to the amazing array of service to her profession, she's maintained an outstanding level of research productivity on landscape level cross-scale ecosystem ecology. Her research record led to her election as a fellow of ESA and the American Association for the Advancement of Science. So with that, I'd like to pass this over to Dr. Peters. Thank you for joining us today.
Great, thank you. Let me see if I get my slides started here. Okay, so what I'd like to do is just provide an overview of one of the projects that I've been working on for just about the last 5 years. I'm a landscape ecologist, but this is a problem that was presented to me. I was actually at a workshop and I saw a person present this, and then I sort of like schemed with him later on how would we actually solve this problem. So it is going to be it's an animal disease primarily in the US that affects horses, and it can be transferred to humans, but it's primarily a disease that affects horses. So our goal was to develop a strategy and a framework for complex problems. That's what I work on. And I got sort of drawn into this disease problem because it was something that the ARS was really interested in, the USDA. So our approach is spatio-temporal modeling of cross-scale interactions coupled with human-guided machine learning. And then we're just applying this to vesicular stomatitis. It's an infectious disease of livestock, as I said, that causes economic loss, quarantines, and restrictions in international trade. So as a model disease, it actually works quite well because we had access to the data through another USDA agency, the Animal and Plant Health Inspection Service. It's a reportable disease. It looks like foot and mouth disease in cattle and pigs, which has been eradicated from the US since the 1920s. So it's a big deal, right, to actually get this correct. So they actually report it to APHIS. APHIS goes out and checks, make sure it's not foot and mouth disease, but then they actually keep track of VSV. There are multiple hosts and insect vectors. In the US, it's primarily horses. It comes up from Mexico. That's its endemic region. That's where it's mostly in cattle. And then there are multiple insect vectors. The outbreaks occur approximately every 6 to 10 years, but it seems like that frequency is increasing. And it is the most commonly reported vesicular disease of livestock in America, so it is kind of a big deal. So when I first saw this figure, and it showed, let me get rid of this. I was at a talk, I said, and I saw this pattern, and the person who was talking about who worked on VS for a very long period of time, especially in Mexico. And he said, we don't understand, right, the patterns here, because sometimes in 2004, the disease, these are occurrence data, the points are, sometimes it'll stop in Colorado, other times it goes all the way to Montana. He said, we don't understand. So this was the question initially, was what explains this spatial pattern in VS occurrence? And they had a lot of data, but they hadn't actually analyzed it in that way. Oops. Okay, so first of all, it seemed to me as I listened to them that we needed a transdisciplinary team. So Dr. Luis Rodriguez was the one who's actually been doing this since his PhD. He was a virologist. They also were working with the APHIS in Colorado, an animal epidemiologist. But what we needed really was this, okay, it's a disease that actually, there's insect vectors, so we need people who actually understand the insects. So I pulled on the people in Kansas for that. There are people who actually then, that still doesn't get us into the environment, right? So we needed the people who connected the disease to the environment, so like a range scientist. So that's our people in Fort Collins with the ARS, Justin Durner. And then as I listened to them, actually some of these insect vectors spend part of their life cycle in water, and it's flowing water. So then we need an ecohydrologist. And so I brought an ecohydrologist. And we've now, our initial team was very small, it was 5. And then we expanded out. Now we're actually working in the endemic region of Mexico as well through Kathy Hanley at New Mexico State University. So the first thing I asked was, where's your systems diagram? And they didn't actually know what that was. So we had to put this together. And so I put the figure together by listening. So what I didn't want to do was to have sort of a correlation analysis, which is very typical for a geographic disease spread, is you sort of correlate, environmental drivers with the occurrence data, because we need to be able to actually manage this disease and understand the processes underlying if we're going to actually do predictions and sort of provide input to management. So I wanted to relate all the information about the processes, which is, whether the insect population dynamics or how they spread it to the host, or how the host then spreads it to humans, or how it goes back to the insects. I felt like we need to understand the processes in order to understand, how to move forward on this. And so this is a multi-scale diagram. The green area is the local scale, sort of environmental conditions at the level of a farm or a ranch. And then the landscape scale is, we can move animals, they can be transported and start an infection someplace else. Then we also insects can disperse. So I consider that sort of the landscape to broader scale and our broad scale climate drivers. So this was the diagram that we put together over about a period of 6 months, just basically bugging the people who actually understood the disease quite well and coming up with this figure. And this then led us to the data that we needed, right? But again, we didn't want to just use the drivers, which is like, for example, soils. What is it about soils? Because soils are actually complex, right? So what is it about soils? So we went through each of those key processes in the previous figure, and those are the ones in yellow. And then we went through each of the major drivers, right? So climate, soils, elevation. And then we asked, so what is it about those? So we never have the data that we need. So we created what we call Ecotransfer Functions, where we work through, well, what do we know? From the lab, a number of eggs per adult, there's related water temperature, for example. And then we work our way through until we can actually get to a relationship that is a hypothesized relationship between some soils variable, here it's available water, and then the density of eggs. And so they haven't actually collected that, but that's now a hypothesized relationship, which tells us, okay, it's AWC is the soils variable that we need, right? Instead of silt or sand or clay, right? So we did this for every one of our processes and all of our drivers to get to the variables that we need. And then we just flip around and go through it again. So precipitation, what is it about precipitation, right? So this is how we actually came up with the variables from our drivers. So soils is the driver, AWC is the variable, and then we could go and get the data. So we came up with a variable we consider a data cube, which is in time and space and variable name. We ended up with 484 of these by the time we actually do all the actual time element. So it's fairly large data cube. And then we ran the analyses. And so we only have presence data for at that time for VS. So we then, after we standardized and harmonized the data, and we did it at a 4 kilometer by 4 kilometer grid cell across the entire Western US, because that's where the VS occurrence is. So we then, after we had all the data harmonized, we could then analyze it. And so because we only have presence data, we're a little bit limited in what kind of analyses that could be done. So we used a MaxCent, which is it's a species distribution habitat model. It's basically a multiple regression across the entire US. And then that then provides us with some really interesting results. So I think we have time to cover some of those results, but quickly, so basically, remember, they basically knew nothing, right? They knew a lot about the viral genetics. They knew like where the occurrences were. They had some ideas about what the vectors were, but then they didn't really understand the ecology, right? So the first analysis we did was this MaxCent modeling. And what really interesting is that we used two outbreaks, the 2004, 2005. The first thing I did was separate those two separate, the outbreak into two separate pieces, an incursion year and an expansion year. And it turns out that different variables are important, but you need all of those variables, right? So you need climate, you need soils, you need elevation, horse density, of course. But it turns out in the first year, the incursion year, it's related, one of the important variables is distance to water. Well, that's actually black fly habitat. That's one of the vectors. And then in 2005, when you look at it, again, it's climate, it's horse density, it's distance to water, but now the soils property comes up. And that was the one that we actually put in there because Culicoides is another, it's biting midges, is another vector. So what the data is showing is that, it's one vector is important in the first year, black flies, and then Culicoides, the biting midges, a separate vector becomes important in the second year. And so it's only by sort of, combining these process-based information with the broad scale sort of climate and other drivers could we actually come up with that analysis and result. And then we tested it, right? We had a second outbreak. We had 2014 and 15, which is a different outbreak, and actually had the same result. The same variables came up. So 2004 and 5 are related, genetically, right? And 14 and 15, but it's actually the environment is more similar. So 2004 is more similar to 2014, 2005 is similar to 2015 in terms of environmental variables. And so it turns out that the environment is actually driving this patterns and not the virus.
Dr. Peters, we have just another minute.
Okay, so then we've gone on to develop early warning strategies based upon this. So it turns out that it's more related to things, in each year, the actual specific variables through time. And then we've applied the same approach to West Nile and Triple E, which is another disease. So I think we're comfortable using this approach. I think the general approach works really well, but in terms of vector-borne diseases. And then we're predicting future outbreaks, right? So we actually 2019 and 20 was another outbreak, which we were very surprised at because we were told it was sort of every 6 to 10 years, and now it's like came after 4. And so we're in the middle, like COVID, and how we actually predict. We're being asked, what's going on, right? How are we going to predict this given what you know? And so we also then sort of using a bunch of different spatio-temporal models to actually make these predictions for 2019 and 20 while it was occurring. And it turns out that, 2019 and 20, you just know what you're doing, and then all of a sudden it's very different. So that one showed up in eastern Missouri, southern Missouri, and eastern Kansas. It had never been that far east before. So it's sort of stretching. I think that what we just heard, you have to be flexible and adaptive, exactly, because these things are changing all the time. So now we're looking at sort of why does it come across the border? Why does it have these different patterns through the different outbreaks? And then, how do you actually then predict these for future? So I think the insights as we putting together a transdisciplinary team, we've heard now a couple times, this is actually really important. Recognizing the importance of processes and how do you relate that to the patterns and not just doing sort of pattern analyses. And then we just have very regular team meetings in order for us to understand and be adaptive. Our challenges are limited data availability. Insect vectors, we don't know very much about their habitat. We have a lot of hypotheses now about their importance, and we're going on and testing those. This information is being given to managers, so horse owners, in terms of what's going to happen. We could happen again this year, so we're trying to make a prediction for 2021. and then multiple computational tools. So we started with MaxNet, we're going to some convolutional neural networks and process-based modeling to complement what we've been doing. I think that's all I had, just sort of our key papers if you're interested. And then we have a story map that on Esri that sort of walks through this in more detail. And then sort of our collaborators here. This is so it's grown into about 15 people now instead of our original 5. And I think that's all I have.
Great. So thanks again, Dr. Peters, for that great talk. So we now move on to our next speaker in this session, Dr. Ruyen Kay from Los Alamos National Laboratory. So his research group focuses on developing mathematical and quantitative theories and tools to understand the spread of viruses, viral immune interactions, and viral evolutionary dynamics across multiple scales of biological organization, that is intracellular, cellular, and population scales. Since January 2020, he has been working on modeling the transmission dynamics of SARS-CoV-2 across the globe. More recently, his work has focused on characterizing within-host dynamics and immune responses to the SARS-CoV-2 infection. Before LANL, he was a tenure-track assistant professor of mathematics at NC State, and he did his PhD at Imperial College, London, followed by postdocs at UCLA and LANL. So let's welcome our next speaker again. Ruyen, over to you.
Thank you for the introduction, and thanks for the opportunity to present our work. Let me share my screen. Okay. So I'm going to talk about estimating key parameter values for novel infectious disease outbreaks and implications for control. I'm going to use my work from SARS-CoV-2 as an example. To put our work into a broader context, I'm showing you the CDC response framework again. Basically, it categorizes different stages of response to pre-pandemic intervals and the pandemic intervals. So the questions I'm focusing on really during the pre-pandemic intervals where a novel pathogen is identified and human-to-human transmission is reported. During that stage, the key question is to assess epidemic potential of the novel outbreak and find out the most effective control strategies to control the outbreak before the outbreak becomes a global pandemic. So two particularly important parameter values for assessing epidemic potential are early epidemic exponential growth rate, little r here, which I will introduce later on, and the basic reproductive number are not. These two parameter values characterize an outbreak in the absence of intervention efforts. So it sets the baseline, therefore essential for forecasting epidemic trajectories, evaluating effectiveness of intervention strategies, and predicting the herd immunity threshold. The last item becomes more and more relevant as of today when the vaccine rollout starts. So let me start with the estimation of little r exponential growth rate. This estimation starts with data collection, and then we fit mathematical model to data to estimate the exponent on the exponential function, little r here. So the difficulty here is really to assess the biases and uncertainties in data collected. This is particularly relevant for novel pathogen outbreaks where we probably lack of diagnostic tools to confirm an infected case. Because of the low surveillance intensity, reported cases may not paint a full picture of the full scale of the epidemic or outbreak. This is well demonstrated by the SARS-CoV-2 outbreak early in 2020. There are other factors, social and economical and political factors are involved in data collection process makes the data even more noisy and biased. So the central question, I think, during a early outbreak of a novel pathogen is how to evaluate uncertainties and reliability of estimation. So due to all these uncertainties, if you look at If you look at the estimates of later R for early SARS-CoV-2 outbreaks in Wuhan, you find vast different estimates. Early estimates were made from groups in Hong Kong and Europe. They suggested the virus grow at a relatively slow rate, 0.1 to 0.14 per day, translating the doubling time of 5 to 7 days. These quantities are important. I want to point out these quantities are very similar to the estimates for SARS-CoV-1. So for a long time period, we thought this virus behaves similarly as SARS-CoV-1. And these works made high-profile papers and have been highly influential in terms of policy making. However, some other groups, including our group, showed that the initial growth rate is actually much higher. The virus is more contagious than SARS-CoV-1. We estimated the growth rate is 0.2 to 0.3 per day. That translates to a doubling time of 2 to 4 days. So just to illustrate the vast difference of the estimates using this toy example, if we assume an epidemic doubling time is 7 days, and assume at time 0 there's 1 infected individual, after 2 months of transmission, we would expect approximately 400 infected individuals. This is a relatively slow spread as we've seen for SARS-CoV-1. However, if the doubling time is 3 days, we would predict after 3 months of transmission, there would be 1 million infected individuals. So actually the latter case is what we experienced in Lombardy, in Italy, or in New York, in the US, for example. So given the different estimates, how can we be sure our estimates are reliable and which one are more reliable than others? So for the past year, the 2 lessons we learned were first, cross-validation using multiple data sets. Each data set has its own biases and uncertainties. So what we think is if multiple data sets point to the same conclusion, that increases the estimation and conclusion, the reliability of the conclusion. So that's one important aspect to consider given the high uncertainty in the data sets we collect during initial outbreaks. Second is predictability, whether a model estimation and conclusion are consistent with future outbreaks. So as I mentioned, we estimated the initial growth rate to be very high, higher than 0.2 per day. So our estimations are different from other groups. So we tried really hard to make sure we believe our own estimates. So we find out 2 data sets that we didn't use for inference. The first data set was the outbreak curve by date of symptom onset published by China CDC. If you fit an exponential curve to the reported cases by symptom onset, you realize the growth rate is higher than 0.2 per day. Another piece of evidence is the curve by death count during late January 2020 from China. If you fit a growth curve, the growth in death count also is much higher than 0.2 per day. So both data sets cross-validate our conclusion that the virus is highly contagious, is very different from SARS-CoV-1. Predictability, as the virus spreads across the globe, we watched closely and estimated the early growth rate in Europe and the US. We find that although there are heterogeneities in our estimates, but in general, the estimated growth rate is higher than 0.19 per day. Again, these estimations are consistent with the estimates we made initially for Wuhan. In particular, for the US, the growth rate is actually very high. This was mostly data from New York. So from the estimation of later R, the exponential growth rate and doubling time, we can estimate R0 value. If we assume a mean zero interval between 6 to 8 days, for the sake of time, there are some complications into which zero interval to use. But for now, for the sake of time, I'll just happy to discuss later on. But I'll just say that assuming this interval we think is correct interval to use to estimate R0, we estimated R0 from Wuhan across Europe and the US, that the R0 value is significantly higher than widely reported R0 value of 3. The R0 value is likely between 4 to 6, which suggests the herd immunity threshold must be very high, maybe higher than 70 or 80%. From the estimation of these two parameter values, the question before the outbreak become a pandemic is really what its implications for control and how best we can control the virus before it becomes a pandemic. So in early February,
just a minute to wrap up.
Okay, sure.
Thank you so much.
All right. No. This is the last results. I'm going to show what we realized, the conclusion we made based on the estimation of a high growth rate, high value for R0, the possibility of asymptomatic and pre-symptomatic transmission. We estimated that test, trace, and quarantine alone wouldn't be able to control the virus, even under the most optimistic scenarios. So early, strong, and social distancing efforts are really needed to reduce the overall contacts and stop the virus. So this conclusion hasn't been well received in the early days of the outbreak. At that time, most people believed that this wasn't a threat to the rest of the world. We can control the virus as we controlled SARS-CoV-2, but data later on shows that our estimation and conclusion are reliable. So just to wrap it up, lessons we learned for a novel pathogen outbreak and how to accurately assessing epidemic potential. First, data probably will be noisy and biased, and the estimation might be highly uncertain. Two ways to test the reliability of estimates: cross-validation with multiple data sets and predictability. These two tests are largely absent from early studies of the epidemic, which may lead maybe policy making are made based on certain estimates. So we can do better next time, probably. And also, I want to mention that there's a difficulty in publishing results that are different from already published results, especially already published results from established groups. I think for novel pathogen, we know so little about the pathogen, and the data are highly uncertain. It is expected to have to reach different conclusions based on different analyses, and it is critical for policymaking perspective to have diverse perspectives for policymakers to make policies based on the best scenarios as well as the worst scenarios. So having diverse perspectives is important. So with that, I would like to thank the organizers for the symposium and the Lano team who made the work possible, and also my former mentor, Alan Perelson at Los Alamos, who taught me about constructing credible models and making reliable predictions. I'd like to thank funding from Los Alamos, SimilarIS, DARPA, and NIH. With that, I'll stop my talk, and thanks for listening.
Thank you so much, Dr. K, for your presentation as well. Been really excited about all of the presentations so far. This has been a great group, and I'm thankful to everyone for the questions that have been left in the chat box as well. So we have been gathering those questions together. And at this time, we would like to invite both the plenary speaker as well as the additional two speakers to be present to answer the questions that you all have presented. So we're going to open up now our panel discussion for some Q&A during the, so we have about 10 minutes remaining. And so Dr. Marathe, Dr. Peters, and Dr. K, if you all would mind just making sure that your video is on and unmuting when you are ready to answer a question. And I believe that we may be joined at times by our session moderators as well, who may be choosing to participate in the Q&A as well. Okay. And so I'm actually going to give the option to the session moderators to open up the panel with the questions if you all would like to. Otherwise.
Thanks a lot, Emmanuel. And yeah, thanks again to all our speakers for really diverse and interesting perspectives and experiences in this important problem. So let me start off by asking one question, I guess, to all three speakers, which I think they all alluded to briefly, which in their talk, but it would be great if they can elaborate. Like, this is to do about success measures. Like, so how do you measure success in such endeavors, right, at the end of the day? I also ask, I mean, I think this is related to the problem of validation, but also in other fields, having clear objective measures of success has led to an explosion of development of, like, say, machine learning or AI techniques to help fill the gaps, right? So are the goals and measures clear in this pandemic space? Do we know, for example, for some of the decisions which people are taking, how do you measure success there? Yeah, and I'll throw this open to all the three speakers because they all talked about this in some form. So maybe we can start with Madhav and then Debra and then move on.
Thanks, Aditya. I'll be brief so that others can comment as well. I think it's a beautiful question and a hard one, of course. In certain specific areas, one might be able to define at least some early measures that you can use. We have done the CDC forecasting challenge for now 6-7 years, all of us, and I think certain measures for pure predictions for a steady state disease, meaning things that happen yearly, can potentially be developed. Even there, we find challenges, right? Every year is different from the other years. We can potentially use other metrics of success, which just tell us, things about how the society functioned well. In the end, counterfactuals are very hard to measure against, in my opinion, and that's something that I would like to, in fact, say that in all of these cases, if we can try and put numbers to the value of taking some actions such that a certain event does not happen, we would be in very good shape. And that's really very hard to do, because if you did very well and that event did not happen, oftentimes it gets unnoticed and it's very hard to justify. So I'll stop there and let Debra and others speak.
I think we were fortunate enough that we were able to do the verification. So we had 2 separate outbreaks, so we could actually do our model development on the 2004, 2005, and then test that with 2015. And so I think that was our approach, was to sort of hold some of the data back, which is a classical, way to do verification. And then, of course, the next outbreak came, and it actually it didn't work, right? So as it changes, right? So I think we have to have quite a bit of data to actually be comfortable about these predictions. And this is a worry about with climate change. The climate continues to change, and how do we actually make that moving target when we have basically we're using our historic information to make predictions about something that's changing, and that's much more difficult to do.
Yeah, success in our field is, I was trained to always saving lives, but it's for pandemics, it's a hard thing to measure, because you can't replicate the pandemic as you do in laboratory. So it's hard to evaluate how many lives one saved. So that's a really hard question. I agree with other panelists. But in terms of a model inference, I guess success, I would define as what I wrote in my slides, where cross-validation and predictability of the model would be in a measure of success. That's what I would think.
Thanks so much. Krista, do you have a...
Yeah, I had a question for Dr. Peters. So given what you found on the drivers of the outbreaks, how might climate change impact those, the outbreaks that we see in the future?
Yeah, that's a great question. And so, as I just said, we're using historic information, but we're assuming that these relationships continue. So what we've done is to use sort of these what I call pattern process relationships. And so we sort of then go back and say, well, how would change the precipitation effect of insect vectors, right? Instead of doing sort of correlations, it's sort of getting back to the drivers and process relationships, and then running our models again. So we're doing those climate change simulations. One of the other things we're doing is sort of we step back, and in ecology, a lot of times we sort of grab a scenario because it's popular in their literature. And we actually have a climate scientist now on staff. And so she's saying, that's not really what you should be doing. So, so she's actually providing that analysis of, for this space, which climate model should we be actually using? So we back a minute to actually say, okay, let's actually do the climate scenarios correctly to put into the model. So that's where we're at.
So thanks, Dr. Peters, for that answer. So this is a question which is coming from the audience, which is, I think, again, pretty general. So maybe it's directed to whoever wants to answer, I guess, on the panel. How do we ensure the needs of global healthcare workers are considered in all of your research efforts? Is that a special, do you need to do some things differently, or it follows very naturally from what you're doing?
So I can start, and others should certainly chime in. I think it's, again, a good question. Various demographic groups are important for different reasons. Medical workers clearly are some of the most important ones when pandemic comes through. At least the way we have modeled, we have tried to take that into account when we build a digital twin of a city or of a state. We have information, not perfect, but certainly we have information about the kinds of activities or professions people might have that allows us to capture certain aspects of it. Now, of course, very precise information about individuals is very hard to get, and probably we don't even want it for privacy reasons. But point well taken, and I think the models that we have built at least try and account for that demographic group quite well.
Yeah, so they aren't actually part of the problem for us, right? So ours is an animal disease. It can be spread to humans, but you just sort of get sick and you're well a few weeks later. So sort of a dead end host. So I don't think that really we haven't had to address that. We do have PII issues with our data, which are sort of related to what you're asking, that we do have to protect APHIS data. If you look online, APHIS is at the county level, and we have access to the actual locations. So I think there are sort of those kinds of issues as well that we've had to address.
So I guess another question would also coming from our audience is, with the public awareness of pandemics as high as it is now, what do you think we as a research community can do that we couldn't have done a year back to significantly push the envelope on the science of epidemics? Again, this is really a question which is, I think, open. So maybe we can start with Ruyen.
Yeah, this is a great question, very broad question. I've been focusing on very specific problems for a long time. So probably my answer would be, I think surveillance is really an important area to focus on, to know what kind of viruses are around in animal reservoirs, because most of the viruses jumping from animals to humans, then and also how they jump from reservoir to human, to understand the human and the reservoir interaction, the interface. I thought that would be a very area that could really help to prevent the next pandemic.
So I can say a few words, Ditya. I think Anand's question is really on point. I think there's a huge awareness right now, but there's already things in play right now. There's a directive from the President's office, as you saw, which aims to establish a national coordination center, and which is going to really change what gets done. Its shape and form, of course, is unclear right now, but clearly is going to happen in the next six months, and that will allow people to come together in a meaningful way. One of the things that may be worth thinking about as a group is to really realize and appreciate multidisciplinary science because of this. This silo-based science is not how this problem is going to get solved. And maybe if one thing I take this as an even stronger signal for me to continue my work is we have to work in a group with people with very different backgrounds and insights and expertise. Without that, I don't think so we can make meaningful progress on any of these problems.
Yeah, I would agree completely. I mean, we pulled together a team that had never worked together before. And the only way we made the progress was by listening very carefully to what everyone had to say, respect what everyone has to say, and then putting that information together into a systems and model, right? So it had to go somewhere. So actually putting it together into a conceptual model that everyone agreed on, and they could see where they fit and how all the pieces fit together. I think that's really important. I think the other thing is we have a lot of data, right? We tend to have a lot of data, and we have a lot of technology, but but I think that we have to then provide that information in a way that people can understand. So I think that's the other piece, especially when we start talking about the public, is how do we actually provide this information so that we do have a lot of results, and how do they get the right information and that they can actually understand it and trust it.
Thank you, everybody. I've got one last question we'll fit in before I think we're going to take a break. This comes from one of our participants. How does social network information across the US consider on the ground of context, especially when it comes to minority communities? So I think this question would be for Madhav. So how do we consider the systematic bias as it relates to our minority communities?
I think it's a good question. I was thinking about it when it was posed. So first of all, I want to acknowledge this real problem that we have in the current epidemic, and in fact, almost all large-scale disasters. A minority community always gets disproportionately impacted, and this time is no exception. So very unfortunate that this is happening this way. But at least in our modeling framework, Krista and the person who asked the question, we have tried to again build a digital twin that tries and accounts for this. So if you look at our counties and representations, we actually have neighborhoods that have folks with those demographics. They do certain kinds of activities that again reflect, to the extent data is available through surveys and other means, the activities they do. So in our digital twin, this effect at least gets captured structurally. And we do see, as a result, impact on these communities that is somewhat different. But I think the question is really an important one. And I think as we go on improving our networks further, any information that can be made available in a manner that is first, speaks to that community, but still is privacy preserving and confidentiality preserving, would be very useful to put in the models. I don't think some models are done by any means. We really look forward to working with folks who can provide us this information.
I believe I reflect everybody's sentiment when I extend thanks to all of you, our panelists, for your remarks. Thank you so much for helping to inform this conversation and for the value that you bring based on your experience and your expertise. I'd like to also thank all of the participants who asked questions and who provided questions in the chat box. I know we did not have time to address every one of the questions that you asked, but I do want to assure you that all of those questions are being documented and will be incorporated into a report that goes to NSF at the conclusion of this workshop. So your point of view has been captured, and we do appreciate all of you for that. So thank you again to all of our panelists and our speakers at this time.
Thank you.
Thank you.
Thank you so much.
I want to take just a quick moment as we move forward in our agenda. We're going to be moving to our breakout sessions next. And so I want to just provide some quick, just a point of process. For those of you who have registered for the breakout sessions, you have received a separate email from NSF, which includes a different Zoom link for the Zoom meeting, which is different from the webinar that you are in now. So if you are going to be participating in the breakout sessions, please make note that you will have to exit this Zoom webinar and use the alternative link that was provided to you via email in order to open up the Zoom meeting. And the breakout sessions will take place then. So we have a few minutes over break. I do encourage you, if you are going to be in the Zoom meeting, please make sure that you enter into the meeting before you leave for break. That will give us the opportunity to put you into the appropriate breakout room. So if you are going to be participating in the breakout session now, we ask that at this time you leave the webinar, move to the Zoom meeting, and do so before you take your break, before you grab your coffee, your water, or whatever it is that you need. For those folks who are going to be joining us here in the webinar, but will not be participating in the breakout session, We will be back here in the webinar for a wrap up towards the end of the day. And that'll happen this afternoon at 2:25, where we will continue with session 2 and our discussion on the molecular level. So thank you, everyone. We're going to break at this time. And for those who are in the breakout sessions, we look forward to seeing you there.